Everyone. Predicting protein structure from sequence is a very important problem, and it's a very challenging problem, as we have learned from serious presentation today. So generally, protein structure prediction involves two steps. The first is try to simulate the models, generate a number of conformations. The second step is to assess the quality of this model. Which model is more similar to the unknown tertiary structure? And I call the first problem is a machine learning problem. Basically, try to generate the predictions, candidates. The second problem is a data mining problem. Basically, pick the best model out of the pool of models. And today, I'm going to focus on the data mining problem. And this problem is called model quality assessment. Basically, assess the quality of those models, and we can classify these model quality assessment methods into two categories, based on two criteria. One is the methodology. For instance, the energy function, as Michael Levitt described in his keynote speech. And also, we can also use pseudo energy, like statistical potentials, or we can use machine learning data mining methods to assess the quality of those models. And we also can classify model quality assessment methods based on the input. We can develop methods that assess the quality of one single model, which is very important because you don't need any other model in order to tell the quality of one single model. Or we can use the consensus clustering-based methods, try to compare the quality of one model, compare the similarity of one model against all other models, and calculate the average similarity between this model with other models, and use the average as an indicator of the quality of these methods. And it's very difficult, this is a very difficult problem, because no single individual methods can really consistently pick the best models or accurately assess the quality of models in all the situations. So then a natural idea is to use a consensus approach. Sounds simple, but actually in reality it's very difficult. I still remember last December after the CASP meeting, I had a conversation with Yang Zhang, the guy who developed one of the best protein structure prediction in the last several CASP competitions. He said to me, which method did you use for the human prediction in the CASP? I said, I used a consensus approach. He said, we used many times, it didn't really work. I mean, sometimes it works better than individual methods, sometimes didn't. I say, the key idea is we need to use large-scale compilation of many consensus individual quality assessment methods, combine them synergistically in order to assess the quality of models better. So here's the basic idea we have. Give a group of models generated by homology modeling or comparative modeling. We use a bunch of the quality assessment methods to assess the quality of each model, and then we try to generate the consensus ranking of these models, and then we go through the refinement of the finally selected models. So here is the methods we selected. Basically, we have 14 individual methods. The blue ones were developed in my own lab, and the other black ones were external methods, including PRQ2, many other methods whose developers are here probably. And the green represents the single model quality assessment methods. For instance, the Marcom Novo used the structure features and the physical features and also the chemical features like hydrophobicity described by the first speaker to assess the quality of each model. And Opus used statistical potentials, and PRQ2 used structure features, and RWPIS also use structure features and the context between the residues. And we also use machine learning methods combine the different information like the secondary structure, context, disorder information even can be used for the structure prediction, quality assessment, and also conservation of the sequence used to build those models. And we also have the energy function-based methods. So these are single quality methods assessment methods. And we also use a bunch of the clustering-based methods to complement these single model quality assessment methods. Then we combine them to generate the consensus ranking. So the question is, how did we come up with this solution? Okay, so basically I still remember a meeting when I had 10 days before the CASP started. CASP is a blind assessment of the protein structure prediction methods in the worldwide. We have Anna and Thorsten, who are the co-organizers of the CASP 11. So the basic idea is, my question I talked to my student, how can we come up with a method that can assess models better than the other predictors like developed by the Keza Han, those kind of guys who have the dozens of years of experience of developing the energy function to pick the models. So we have no clue about how to do it. So then we just come up and say, let's pull up large scale, do the large scale consensus of those individual methods. So we select all the methods available before the CASP 11, and then we bunch up these methods together on the CASP 10 models. And actually we find that combination of 6 methods performs the best. It's slightly better than the all 40 methods. But we are afraid that this might actually overfitting the data. So we still keep the 14 combination of all 40 methods. And then we also decide to use more single model quality assessment methods because clustering-based methods tend to bias against the redundant models. Those models are very similar to each other, but could be of poor quality. So we use more single model quality assessment methods and with some clustering-based quality assessment methods. So here is the general paradigm we're using. Given all the models predicted by the CASP server predictors, and then we first use individual methods to assess the quality of these methods, then generate consensus ranking, but we do not stop from there. We also clustering those models into categories. We try to pick the top models from different clusters. I'm going to explain you why. So, and then we select the top models and then refine them as our final predictions. So here is how we combine those methods. So given a number of models, about 200 models for each target, we use individual methods to assign a quality score for each model. We also can convert the quality score into Z-score, relative score. And also we can use that score to rank those models. Now, we have 3 different ways to combine them. One is we can just simply average the score of each of that model generated by different methods in order to rank them. Or we can average the Z-score to rank them. Or we can average the ranking of each model to rank them. And in our CASP experiment, we use this ranking approach because it makes the predictions score on the same scale. It's easy to compare. Once we get the consensus ranking of all the models, then if we just simply take the top 5 models, they could be very similar. If one fails, all 5 will failed. So we use a statistical approach called stratification. We classify models into different categories. We always select the top 2 models ranking by the consensus ranking. We trust them. Then we select another highly ranked model from other clusters, different from the cluster of the top 2 models, in order to increase the diversity, in order to make one of our top 5 models would be better. Here are the results. On the CASP 78 protein domains, also CASP 11 experiment. So our method ranked 3rd among 143 predictors. I think even though it's not the best, but I think it's significant. Why? I explain why. Because this method is largely automated, and this method can achieve the performance on par with the best methods involves best brain in protein structure prediction, like Yang Zhang's brain and Jianlin's brain. That's highly intelligent methods developed by those people. And also better than 140 other methods. So that's significant. Now, I also compare our method against the server predictors that generate these predictions. So we can see that this method, this is the sum of the Z-scores of the top-ranked models selected for those domains is better than the best server, Zhang server, and many other servers, including our multi-count cluster, construct, and Lenovo, those top servers too. Now I try to assess the contributions of individual quality assessment methods in this group, in this consensus approach. So the top three are our three combination methods, including the multi-count ranking-based, average score-based, average z-score-based. So other individual methods, we can see the average score of the top one model selected by these methods. So the first one is better than all other ones, including the average z-scores of the top models selected by these quality assessment methods. We can see these consensus methods are better than those methods. In terms of then we do the statistical significance test. And we compare this consensus method against each individual one. The difference is significant. And we also try to log out one single method to see what's the impact, right? We look at the individual one, we find if we remove this method, the score drop most. That means this method could make a significant contribution in that consensus. And many other methods actually also make important contributions. Now, I would like to show you a few examples how this works. So here is one target, 855. This is the distribution of the GTT scores of all the models. It's a bimodal distribution. Here is the plot of the GTT scores of this model, real GTT scores of this model against a ranking from rank 1 to 200. So we can see there's some negative correlation. That's a good thing because highly ranked has higher score. And in this case, we select this model that is best and the blue structure superposition with the model gold. And the score is 0.6 around. Here is another good example. Distribution of the scores of these models by model. There's some good models, there are a lot of bad models. And this is the plot. So this model has been selected and also is one of the best models. Now I'd like to show you one example we failed. So here is, for this prediction, it's very hard case. You see the distribution of the scores in the range 0.0 to 0.6. There are 3 good models, very, very good, but the majority of them are bad. Here's the plots. We can see we select this one, rank 1. Actually, the best is here. You see the topology superposition, blue structure, gold model. Both are helices. There's no difference in the secondary structure. But this is more compact, gold one. So most methods prefer more compact structure rather than extended ring-like structure. So all those methods have been fooled. That means some information is missing here, because in nature, most proteins are globular. So they don't like this extended structure. So all the methods have been fooled. That means we need more methods in order to discriminate this kind of the situation. So with all this work, what we have learned since last year, last April till now. So I think we learned a couple of things. Large-scale quantity assessment method works. We need more and more of these such methods to add together. Hopefully one day we can really significantly address this problem. We can select models, with very, very high quantity, high accuracy. Secondly is the way to combine different quantity assessment methods matters. Otherwise, it may not work. Then we may question the whole consensus approach, whether it works or not. Actually, indeed, we show that it works. And single quantity assessment is complementary with the clustering-based approach. And I believe it's more significant, it's more powerful, could be, because they are different. Clustering-based approaches are generally the similar approach. So, and with that, and also we can construct a web server called a multi-count server that for you can make structured predictions. You also can use our server, use this large-scale quantity assessment to assess the quantity of models you generated from other servers like Zhang server, Rosetta, those kind of the top servers. With all that, I'd like to thank my students, Renzi Cao, who actually did most of this work with help from other 3 PhD students, and thanks the NIH for the funding, for supporting this research. I also would like to thank the anonymous reviewers. That's the first time I thank the reviewers in a conference presentation, because he not only accepted our paper, most importantly, he gave us very good idea to use the other 2 consensus approach, average z-score, average z-score to rank models. So that's a post-analysis after our own experiment. I really appreciate that. And thank you very much for your attention.
among the methods to describe to calculate the consensus, you have several highly correlated methods like dope, d-fire. So it would be better maybe to have a small amount of methods, but they are giving different aspects of the assessment?
That's a great question. Yes, redundancy causes a problem. You combine two redundancy model, actually doesn't help. In some time, actually reduce the assessment. So there's certainly, I believe there's some redundancy between those methods, but also largely complementary. That's why it still works. We haven't got a lot of time to really do a serious correlation between those things. That's something we need to do. How do you add methods into your consensus approach? Try to avoid the redundancy, but it's impossible to completely remove redundancy because all these methods use some similar features to some degree. They only have a little bit correlation. As long as they can contribute some new information, that method can be used.
I was just wondering if your models or your method allows you to study models also at the local level rather than just the global level, and what your method is saying about that.
Okay, that's a great question. Okay, that's another way to think about the quality assessment methods. One is global quality. You predict one single school about the topology of this model. Or local quality. You assess the quality of individual residues. Actually, all these methods have been developed. For this purpose, we only, because that's for the CASP, and also we only care about the general overall score, so we use global quality assessment. Actually, there's a lot of methods, including us. We have developed the local quality assessment methods. They are available. They're very important because you can assess the quality of the key regions, like a binding site, how those regions have been predicted. They are available. Another contribution from the local quality assessment methods is you can pull the local score into a global score. So that's, sometimes we do those things, actually, in this consensus. So that's really a good question.
One last question. It seems that you don't have any solvation terms among the features, or do you have some terms that describe the...
Oh, solvation terms. Okay, that's a great question. Let me look at whether or not this method considers a solvation. Okay. Maybe this method, Select Pro, actually Select Pro may consider some solvation energy into its function. So, because it learns some terms from the raw data, those kind of the energy function, so could be used. But I haven't listed those, specifically checked the individual things. Yeah, that's another thing. If it's missed, it should be added here. So that's a great point.
Yeah.
Yeah, hydrophobicity, hydrophilicity.
But it's a pairwise potential. No?
Yeah, it's not the best way to represent the hydrophobicity, but it's there. Hydrophobic residues very much like this one.
Okay, we thank the three speakers of this morning session. Thank you.